Hello.
Hi.
Hi, how are you?
PowerPoint.
No, I didn't check yet. Is this like something you just sent me?
It's just because you were describing brain development and it's due to crawling cells. I went, oh, well, that's an explanation for why things don't develop in microgravity. It's just one of the things that goes wrong. I could show it to you if you want.
Yeah, that'd be good. Why don't you go ahead and show it? Hello, Barshan. How are you?
Yeah, I'm good, sir.
Good, thank you.
So, I worked on that Eigen embryo thing. So I got a data set about cell tracking.
Okay.
It's a bit of an old data set. I also prepared a presentation for it, and I also have some code and results. So I can show that once we have time for that.
Yeah, we'll do that after Susan. Susan's going to present
Yeah.
something, and then we'll let you present. Okay.
Okay, sir. Okay, sir.
All right. Well, yeah, looking forward to it.
Yes, so me too.
Oh, yeah. All right, I've got it. I'll share my screen, and then you can talk about it and do that.
Okay. So since we were talking about brain development, last week and it involved crawling cells, I went, aha, it's one of the reasons why things don't develop in microgravity. So anyway, so I made a slideshow. So okay, well, this is just simulated microgravity, and actual microgravity is worse. So it alters morphology and cytoskeleton organization and cellular mobility and cytoskeleton protein expression. And this is in rat hypothalamus, but it happens everywhere. And this is an example. Mind you, the cells usually recover within 24 hours. You can adapt to microgravity, which is really great. But if you're a developing organism, it doesn't work so well. So this is what these cells look like. You're looking at the microtubules in this cell, and this is a lymphocyte. It's an immune cell in the blood, and it looks like this in microgravity. And these are all disorganized.
So it's disorganization is the effect.
Yeah, and this is a summary in a paper I wrote, and there's these. If you look at the bottom, these are cells in normal gravity, and then there's cells in simulated 1G, and then there's cells in microgravity. So there are differences. If you have cells in space in microgravity and you spin them, they even look different from those in normal gravity. And this is in a developing organism. So I think it was a rat pup ear or something. Anyway, it changes. They change configuration depending on their environment, their gravity environment. And if you look at the next column, you can see that at the bottom, what microtubules, I believe. Yeah, they're microtubules on Earth, and it's a very rich pattern. And then if you go up, you get microtubules in simulated microgravity. So you're starting to see the problem here.
Yeah.
But there's without vibration in A and with vibration in B. So even vibration will help the microtubules grow in. And then you get microtubules in simulated 1G in space. And one's the side and one's the top. And you can see that the pattern there is not as rich as the microtubules on Earth. And then you just get microtubules in space microgravity, and there's about nothing there. It's just not there. And then you look at the differences in gene expression, and you get in Earth gravity normal, they're clustered. And if you go into microgravity, they spread. They're more spread out. So it even affects gene expression. Okay, next. unless you want to see it. Okay, so I just zoomed in on the microtubules in normal Earth gravity, and then in simulated microgravity, and in microgravity, but with simulated one gravity, and then microtubules in microgravity. So you can see that better. It definitely affects microtubules, which is a major part of the cytoskeleton. And they've even got one for microtubules in magnetic fields, which is interesting.
Oh, what's a magnetic
the same
field?
Authors.
Like, what's the difference between a magnetic field and gravity?
I don't know. I'll just say I don't know the exact things, but someone did an experiment, and I can't find it, but they did an experiment where they spun these flasks in a magnetic field in microgravity, and it restored the patterning.
Okay.
So, yeah, and they also did it in high magnetic fields. And if you have too high a magnetic field, like say 7 Tesla, you get the microtubules align too much.
Okay.
Like they align a lot. So that has something to do with MRIs.
Right. Yeah.
So Nick did an experiment with a 11.7 Tesla magnetic field in an MRI with salamander eggs. They all died, except for the ones that were about a foot away, and they showed extreme defects. So 11.7 Tesla, don't ever use it in an MRI. That is not a good idea.
Well, I think for small animals, yeah, it is. It's what I've heard. Yeah.
Yeah, no, it does affect the microtubules.
Yeah.
And if it's a developing animal, it's not a good plan. But what is it? Use 4 Tesla maybe in MRI. It's not.
Right.
Anyway, it's a J curve again. You can go up to 6 Tesla or something, and there's really no effects. But then it takes off.
Right.
So by the time you get to 11 Tesla, it's like, oops, shouldn't have done that. Anyways, that's just an aside. Anyway, next slide. So these are some salamanders that they sent into space, Mir space station. And salamander A there, I'm not sure what to say about it. It was taken up to space at stage 55. And so it's a bit weird looking, at least to my eyes, because I'm used to looking at salamanders. And then B is the one that they sent up into microgravity. And C is the ground control. And if you look at the eyes, really zoom in on the eyes, you'll notice that B looks cross-eyed. I took one look at it and I said, that salamander is cross-eyed. In other words, I believe it has microencephaly. But I don't think they checked that. But that's my thought on it. Okay, next slide. I hope there's a next slide. Okay. So reduction in gravity leads to reduction in the pre-stress of the actin network with neurons. Spaceflight reduces the actin fibers in human monocytes. Okay, I don't know. And a few examples of astronauts suffering from schizophrenia spectrum problems during or following missions. Anyway, so there's an effect on the brain and on the immune system. And I don't know if is there a next slide? Did I manage? Next slide. Oh, okay. So crawling cells in brain development, migrating tangentially and radially. And if they don't migrate, you get microencephaly. So what happens in microgravity? The cytoskeleton isn't working as well as it should for various reasons. And you don't get the cells placed where they should be because crawling cells have difficulties in microgravity, like I said, including immune cells. And so I don't know, is there a next slide or? I forget. Oh, okay. Well, I circled the crawling cells here. So you need those. You need them to crawl where they need to be in order to create a decent brain. So I suspect the salamanders that were in that flight were not normal, as they claimed at first, but that they had microencephaly or something else going wrong with them. Anyways, I guess, is that it? Yeah.
Yeah, basically that's the takeaway.
Yeah, that's just it.
Yeah. So microgravity alters cytoskeleton and will alter migration of cells. All right, that looks great. Thank you, Susan. Yeah, it's always fun to look at some of these results. Yeah, so a magnetic field will restore cytoskeleton up to a point, and then it destroys it.
Yeah.
Okay.
You have to get it right. You have to hit the right spot.
So is it like that? It's like a field that's realigning it, and then if you have too much of it, it just kind of overwhelms.
It really aligns the microtubules and keeps them there. And there's a cancer medication that does the same thing, and people lose their short-term memory while they're on that drug. Yeah. And it's the same idea. Cells that are reproduce a lot are stalled by aligning the microtubules. I used to know the name of it and gave it to my sister. Going, she says, yeah, my short-term memory is gone. And I went, yeah, well, they're giving you that drug, yes. Anyway, she recovered. My sister's brilliant and normally doesn't have these problems with her memory. Anyway.
That's great. Thank you, Susan. Yeah, thank you. That was great. So yeah, any other thoughts?
Okay, so after this presentation, I had a few questions out of curiosity. So after looking through microgravity, I had this question: does it anyways speed up the growth of the organism, or does it only affect the sequences, like the gene sequences and the protein sequences? So does it just lead to a kind of mutation, or does it also speed up the growth in any ways?
In some things, it speeds up the growth, but it alters the cytoskeleton, which will alter the gene expression.
Okay.
So it's not a good thing if you're going to be full-time in microgravity, you need to at least have a rotational field of some sort to restore.
To cancel it out.
Restore it. Yeah, well,
Okay.
yeah.
Right.
People don't belong in microgravity.
Yeah.
Yeah, so we have to reproduce 1G.
Okay. Thank you.
All right. Yeah, that's great. So Barshan, do you want to go ahead and present?
Yeah, sure. I'll just share my screen.
Here, I'll turn everything off here.
Okay, so I'll just recap on what I ended with last meet. So basically, I was thinking about this idea about why don't we use this Markovian dynamics of breaking the movement of the organism down into eigen movements or vectors and use this same thing for an embryo. So I got a data set in which I could operate on this and do some analysis. So my problem statement here was that the embryo consists of like a lot of cells and all of them are moving in 3D tracking. Each cell individually is very noisy and it does not capture how each cell is correlates with the other one in a global manner. So basically there are a lot of dynamics also including in this. So there's in one way there is this elongation which is like the trend of its growing, and the other one is this fluctuations which is due to like the changes in its muscles or like the interconnecting network of proteins which is called like the cytoskeleton of the embryo. And so we need a low dimensional representation to like analyze on this development. So next, this is the data set which I worked on. It's like a cell tracking challenge. It's quite an old data set, so I had some problem with it. It's like 2008 or so. And it had the 3D coordinates of the cells over time, like in XYZ, and it was extracted from the TIFF mass. So this is the overview of the data set, a few of the frames. So it goes from the like the starting of the embryo till the formation of its the skeleton or the overall shape of the embryo. So how do we analyze this growth? Like how do we treat this embryo? So we take this as a cloud of points or cells, and we take a center of mass and we shift the coordinates to those, and we construct a gyration tensor, which is like a matrix, which is used to capture the spatial distribution and the size and shape of each cell. And then we extract the principal components of each of its shape, and then we analyze upon the growth sector and the microdynamics sector, which is like the fluctuations and the small twitching. So this is the eigen values which I extracted, like the primary length of the embryo, the width of the embryo, and the depth of the embryo. And this is the eigen space, which I created using only the length and width because the depth of the embryo was not changing a lot. So as you can see, it's fluctuating a lot in the initial stages. And later it's like it's normal, but it's fluctuating due to its micro dynamics. And we see this like we divide this dynamics into two steps, like microstate and the macro state, which is like the slow growth or trend of the embryo, and the microstate, which is like the muscle buildings or the cytoskeleton building of the cytoskeleton. So we have this twitching, and this is a dynamic simulation which I code through. And on the left, you can see the embryo which is growing, and how on the right the microdynamics is decreasing as the embryo is growing, since it's getting a shape basically, and it's reaching its final form. So the muscle twitching is getting less. It's mostly into the growth part in the next part. So this is just the microdynamics graph, where in the initial stages the muscles is building a lot, and at later stages we have found the overall skeletal or the shape of the embryo, and the muscles are not building that much. It's mostly growing into a worm. So this is the behavioral stage which I've used using clustering or k-means, where I've separated it into four shapes basically, with early bean, comma, the 1.5 fold and the 2 fold plus. And this is also a center of mass based 3D plot, which I made, where the blue dots are the cells you can see which are increasing, and the yellow arrow is the width, and the red arrow is the length of the embryo. So according to the center of mass of the cells, the width is also changing. And I've got this till 194 frames. There were like 1,000 frames in the data set. So, like a quick recap of what we got out of all this. So basically, I got all the 3D cell data into easy numbers, which is the same thing that the Markovian dynamics did. It separated the movement of the worm into this eigenvectors to analyze it better. And then also a clear separation between the slow growth and the fast twitching, so that we don't mess up the normal trend with the muscle twitching or the cytoskeleton creation which is going on. And I also mapped some plots about the 2D shapes and how the embryo is going through its development phases. So I also deployed this as an open source in the GitHub, and also the data is available. I'll just put this PPT up in the Slack if needed.
Okay.
And this is a future what I plan to do. Like, I plan to go for a better data set. The thing is that this data set was like I made this locally on my device.
Right.
So this data set was the smallest I could find.
Yeah.
And the new one were like 50 GB and stuff. So it was difficult for me to run it on my laptop too.
Yeah.
Like, I couldn't properly. And also I read a paper about how the ages, like there are some latent variables, hidden variables, which also matter during the development phases. So I also thought about, there was a paper in which it showed how the aging of the worm affects the inner working of the worm. So I also thought about how can we predict the age of the worm based on its behavior, if we can reverse engineer that. So this is one of the ideas I also had. And so I'm open to any kind of questions or suggestions that I could use. Like, I'm looking for a new data set or also a new approach which I could go through. Yeah, that's all.
Okay. Well, yeah, you might want to reshare your screen because I have some questions about.
Okay. All right.
But yeah, so the 2008 data set is usually the one we use, and because the cell tracking is basically it's probably good enough, and like you said, the new some data sets are really large, so it's hard to work with. But
Exactly.
so I mean, basically, you want to have the tracking data, you want to have this normalization step. If you go back to the first graph that you showed, I think it was this one here. So I couldn't see the axes. What are you doing in this graph?
Basically, the axes are the length and the width. So it's checking how the length and the width is changing over time. And at the top, you can see it's a lot of points are over there. So it's like clustered over there. It's basically when the worm has got its shape and it's not twitching a lot, because it has already formed its muscles, and the embryo is like developed properly. But in the initial stages, it's twitching a lot and it's changing its shape because of the muscle buildings, or you can say the proteins are getting increased in number, or the network is changing.
Yeah. So this is tracking a cell position in 2D, or like length and width. And then this is like reducing it. So this is like over time, you're tracking all cells or one cell. What does this represent?
This is just one cell.
Oh, okay. So we don't know what cell this is, or we have.
Yeah, it was like a set of cells, and I just tracked one cell, and like this in this you can see like how I tracked one cell and how it's changing.
Yeah. So this is just over time. So it's like the things moving around. Well, I mean, if you normalize it, then there is kind of the aspect of migration. If you don't normalize it, then you're just picking, in part, you're also picking up movement artifact and differences between frames and things like that. So that's, and then, so this is just the I guess, in this data set, it's like it's a centroid that's represented by the fluorescent signal. So we think it's mostly in the nucleus, although sometimes it's not always localized in the nucleus. But basically, it's some centroid we can define in a cell. So that movement, of course, is nice to see. I don't know what the. If you go back to that original graph,
Yeah.
Sorry.
Okay. So it looks like you have like, kind of almost like what they call levee walk, which is where you have some Brownian motion in there, but then it moves to
Yeah.
different places and then kind of explores that place. I don't know if that's just kind of maybe an artifact of the data or what, but that's kind of interesting. But this is
it is.
Yeah.
Just a brief comment. The twitching, that's cell division, isn't it?
Could be.
Yeah, that is cell division. Yes. Yeah.
Okay.
So I would assume that this part up here at the top is cell division because it's kind of in one place, or
Yeah, that is.
okay, yeah. I mean, that's just interesting. I don't know. I would not interpret that, but because it's the eigenvalue, it's not the actual coordinates. So it's pulling.
Yeah,
out
it's
the
eigenvalue. Yeah.
Yeah. So the next slide. So this is okay. That's the actual.
So that's a formula of
the residual.
when we are removing the macro movements from the micro, like the micro from the macro movements, basically.
Okay.
So there's this strength of the growth of the worm, and we are removing that from the actual trend. So we are we are removing the small fluctuations that we get in between. So those we are categorizing into twitches.
Right.
Twitching. Yeah.
Also, it's probably not muscle contractions per se. It's a better way of saying that is the actomyosin activation.
Yeah.
Okay, I'll note that down.
Actomyosin complex, and it's not considered muscle. It's sort of the same.
Yeah.
But
it's what I figured. I just interpreted that it might be like a muscle contraction, or I got it from the web. That's what it can be. So I just put it on the PPT.
Well, it does have a lot of
replace it with actomyosin complex or actomyosin.
I have noted that down. Yes.
Okay.
And then slide 8, I guess. Yeah. So this is from the data set. These are the cell centroids, the fluorescent signal, and then we have this. This is is this a simulation or is this like the microscopy image? I guess it's probably like the movie or
no. Basically we had it in form of frames, so I just put it all together and I made the plots run dynamically along with it, so we can see how the raw data changes and how we can see the behavior of the cell as well.
All right. So the two graphs here, microdynamics at the top and behavioral state space at the bottom. So microdynamics is time, of course, but then what's the y-axis?
The y-axis is the pixel, like how much it's contracting or expanding.
Okay.
Based on the pixels, yeah.
Right. Yeah, it's the. So what's contracting or expanding?
Like the whole we have moved our cell to the center of mass, like the 0, 0, 0, right? Okay. So we can know how it's changing, like the boundary of the cell, how much pixel it's moving, like it's expanding or it's contracting, or how much it's fluctuating.
Okay.
Based on that, yeah.
All right. What do you think that is? Like, is it what would you attribute that to?
Okay, so in the first part of the plot, it's fluctuating very much. So I'm thinking that because it's in its initial stages, it's not able to grasp the whole shape of the embryo, and there's a lot of protein are being synthesized, or we are having this cell divisions which are occurring. So I guess it's it's very much fluctuating in the initial stages, but at the later stages, a lot of cells have formed, and it has achieved its basically its resultant shape. So it's not fluctuating that much, and it's moving towards the trend of the growth.
And then and then you get like at the beginning you get a lot higher dynamics, and then it settles down. This looks like.
Yeah,
Okay.
that's what this. That's what
So is it?
I got.
Yeah, I wonder if it's cell division related as well. Like.
Yeah, I'm not sure about what other components are there, but
Yeah. I mean,
it's what I inferred initially.
Yeah. All right, and then behavioral state space is like a k-means.
It's clustering. Yeah.
Okay. So then that's length on the x-axis, and then width on the y-axis. So it's the sort of the space of the embryo, and then you get the state space. So what is the state space exactly? Is it?
So along with time, the whole shape of the embryo is changing. So initially, it's like a bean form. So like for the first 5, 6, or 10 frames, it's like in a bean formation, and then it's gradually changing its formation, and finally it's getting a secure shape, or like a stagnant. It's getting stagnant over its shape. Like it's a circle, as you can see. So we can say that like we can divide this into 4 stages as the embryo is growing, and this is only the first 150 frames, and after this it elongates as well into a worm shape. So yeah.
Great. And then what's the next slide? I can't remember. I want to see it.
Yeah. So
yeah, this is it. Microdynamics. This is time. And what is the label on the y-axis say?
it's the deviation from the trend, like basically the pixels, as I said. Like how much from the trend it's not moving as the trend should be. So basically it's subtracting the usual movement from the trend, and we get the fluctuations here.
Okay. Yeah. So it's almost like a detrending or something like that, where you're separating out the trend from the noise or something like that.
Yeah, we are separating the noise from the growth. So we are labeling the noise as this fluctuations or twitching.
Okay. And then you've decomposed it into length, width, and depth. So these are the 3 spatial dimensions. Okay. So it looks like width is like basically the largest component of variation here early on. So you have the width. Okay. That's interesting. Well, because we have divisions that are left and right. So a lot of times the cell will divide. You'll have a left pair and a right pair. So they have to divide out laterally, basically.
That's also there. Yeah.
Yeah.
So it takes a big role in this kind of divisions.
Yeah. Next slide.
It's the same slide about this, but this is the final stage what we have.
Okay, so you've labeled the comma, which is around 200 to 300 cells, the bean, which is early before 200 cells, and then the what is that, 1 to 5 fold, or what does that mean?
Yeah, like this is something I got from the internet as well, like about labeling and this embryo when it's completely staged. So some papers it showed that it's labeled as 2-fold or a 1.5-fold.
Oh, okay.
Like since the worm is like gets folded when it's completely an adult form, so I guess it's something related to that.
Oh, right. Okay, I know what it is. All right.
Yeah.
Good. So you've been able to sort of label these clusters in that way.
Yeah.
All right. What's the next slide?
Okay, this is a 3D representation of how the cell is growing and how the center of mass is like changing, and that's where the width is also taking, like the arrow is also fluctuating because of the center of mass of the cells.
Okay. Yeah. And then the next slide.
Next slide, I have a recap and future work.
Yeah. Well, that's great. Thank you. Yeah, and then your
Yeah, I
code
also have it
availability.
available as the code is also deployed publicly.
All right.
If anyone wants to check it out. Yeah.
Yeah. Yeah. The data sets, I mean, that's probably the best data set to work with, is the 2008.
Oh yeah.
They're higher resolution cell tracking data sets.
Yeah, difference,
They're
yes.
harder to work with, but and then we do things like, sometimes we'll want to reference the basic cell tracking. Sometimes with higher resolution images, so you could take like specific time points and take the higher resolution data set and analyze that.
Yeah, if you want to analyze a specific time period of the worm. Yes.
Yeah.
Okay.
Okay. Yeah. Well, thank you. Yeah, that was great. That was a great bit of work.
Yeah. Okay.
So yeah.
Okay.
So yeah. So you could keep working on it. I don't know. I look forward to seeing what you have.
Yeah.
All right. Yeah, it's great.
Yes, sir.
Okay. So thank you to Susan and Barshan for presenting on their things. I'm going to do a paper now, I think. So I've talked about this before, and I found this paper on the physics archive. It's biophysics. This is called Roadmap for Condensates in Cell Biology. So I think we've talked about condensates before in cells. So condensates, we have the cell cytoplasm. And in the cytoplasm, of course, usually in textbooks, you'll see that it's this clear liquid, almost like a jelly. And that's the way they portray it, because you can see through, the membrane, you can see the different organelles and things like that. But in reality, that membrane or the cytoplasm is very packed with things. And we've talked about how crowding in the cell can play a role in function and all of this. So it's much different than kind of what the textbook view is of what the cell looks like inside. So a lot of times you'll have a lot of proteins, you'll have a lot of RNA molecules, you'll have a lot of things moving around in there, and it's a pretty packed place, pretty packed environment. And so, some of these attempts to model single cells has focused, explicitly on that packing, that sort of packed nature of the cytoplasm. And so condensates are these structures that form, they're usually aggregations of like proteins or aggregations of other things, other biomolecules in the cytoplasm. And so this is actually a paper. It's about like a meeting that they had on this topic. It's become a hot topic. I think I talked about a few papers that have come out on this. But basically, people are really interested in condensates. They're interested in the function of condensates and so forth. So there are a lot of authors on this paper. I'm not going to go through who all is on it, just to say there are a lot of people on it. So the abstract reads, biomolecular condensates govern essential cellular processes, yet elude description by traditional equilibrium models. This roadmap, distilled from structured discussions at a workshop and reflecting the consensus of its participants, clarifies key concepts to researchers, funding bodies, and journals. After unifying terminology that often separates disciplines, we outline the core physics of condensate formation, review their biological roles, and identify outstanding challenges in non-equilibrium theory, multi-scale simulation, and quantitative in-cell measurements. We close with a forward-looking outlook to guide coordinated efforts towards predictive, experimentally anchored understanding and control of biomolecular condensates. So this is where, we're kind of at the sort of the dawn of this field. They did a PubMed search where they looked for phase separation and cell as the keywords, which is sort of the things that describe condensates. And this indicates a growing interest in using the concept of phase separation in cell biology. You can see up to 2010, it was really a trivial thing. I mean, when you use these keywords, they don't always refer to this sort of field of condensates, but basically there was very little interest until about 2010, and then it ramps up especially after 2020. So it's a fairly new area. So recent studies have highlighted that condensates are involved in a wide range of essential processes, including gene regulation, signal transduction, metabolism, macromolecular processing, signaling, and stress response. And then condensates are also linked to human diseases such as neurodegeneration and cancer, and even things like carbon fixation in plants. So, it's really a broad sort of spectrum of things that these things are relevant to. To fully understand how cells operate, we need to understand condensates, which in turn will enable us to target or engineer them. The concepts of phase separation and condensation offer a powerful framework for understanding these non-membrane bound organelles. So they're considered organelles. They're not bound to the membrane. They're freely floating in the cytoplasm, providing insight into how they organize biomolecules in space and time. So they're not just these random clumps of things. They may actually be very much functional organelles. And so this is an interesting kind of take on it, because a lot of the things that we've read before kind of give the impression that they're these clumps of things that contain a lot of nice biomolecules, but is it really doing anything in a coordinated way? Is the function coordinated, or is it just function because there's something there and it's sort of an aggregation of useful things to the cell? So we need interdisciplinary science to sort of unravel what exactly condensates are doing in the cell. So the concept of condensation has deep roots in physics. So condensation is where things sort of form from a liquid, so they precipitate into a solid. This is, you'll see on a window, sometimes when it's really humid, water molecules will condense into liquid water. You'll see water there. That's condensation, purely physical condensation. And then biochemical condensation is where you're getting things that condensate from the cytoplasm. And so that's how they're kind of thinking about this. So initially developed to explain phase separation and thermodynamic equilibrium, these concepts have been continuously refined to describe more complex situations in soft matter physics. Similar principles were invoked in biology long before any molecular details were understood. So early observations of cellular space hinted at numerous liquid-like self-assemblies without membranes. So we have these self-assemblies that come together in the cytoplasm, and people saw these and didn't really know what to make of them. Advances in molecular biology and imaging have since revealed that the complex composition of dynamics of these structures, bridging phenomenology with underlying mechanisms in increasing detail. So this figure 2 is sort of a map of the historical development of condensates and cell biology, thinking, back in, the history of physics, soft matter physics, but also in cell biology, and how they've kind of come together since the 1990s to address this question. So in soft matter physics, we go to soft matter physics first. We have van der Waals, who looked at the physics of condensation. This was back in 1873. Coacervation, which was Bungenberg, de Jong, and Kroot in 1929. Then thermodynamics of polymer solutions, Flory and Huggins, 1942. Then complex coacervation, Overbeek and Vorn in 1957. So there was a fair amount of soft matter physics done on condensation and polymers and things settling out in polymer solutions. In the thermodynamics, because it's important to know kind of, in the condensation of water and glass, there's a thermodynamics there. There's a temperature gradient, and we have this thermodynamics that's interesting. Maybe it's trivial in that case, but it's interesting in an environment like the cell. Then on the cell biology side, we have the structure of protoplasm, which was in 1899. That was Wilson. Oparin published The Origin of Life in 1938. Then that was kind of like where people were looking at this, but they didn't really understand the physics of it. They just understood that these were interesting structures in the cell. And then in 1995, you have phase separation in cytoplasm, Alter and Brooks. This is I take it, the first attempt to sort of merge the soft matter physics with the cell biology. In 2007, nuclear pores as hydrogel, Frank Gorlich. Then we had in 2009 P granules, Brangwyn and Hyman. And then in 2012, SH3 PRM FUS, Rosen and McKnight. So I'm not really familiar with, I know the P granules work, but I'm not familiar with this work here. So the future directions, going into the future, looking at complex supramolecular organization. So, what happens at, when you have a lot of molecules at a very high density, what do they do? What is the complexity there? The dynamics and control in living cells. So, do we understand all the signaling implications of these condensates? Do we understand how it exists in the sort of the hierarchy of signaling systems in the cell? And of course, we also need to be more explicit in linking physical organization to biological function. So what are the consequences of thermodynamic fluctuations in the cell? How does that affect things in the cell membrane? How does that affect things in terms of transcription and translation, for that matter? Because we know that those things are highly regulated, and a lot of times it's based on what the cell needs at any given time. We see these studies where people will look at gene expression, and they'll look at, like, if you probe the cell, if you raise it in a certain environment, or you give it a drug treatment, or you knock out another gene in the network, it changes the regulation of that gene, of another gene. So what is the signaling there? Because remember, these condensates in part are the products of aggregation of different macromolecules. The macromolecules ultimately come from gene expression, where they're produced at a certain rate. And so if there's some signaling there that tells the transcriptional machinery to make more of something or less of something, that would be an interesting thing to know. Then there are condensates, of course, in development, evolution, and disease. They didn't really talk too much about development, at least not yet in this article, but they do have some interesting things. I think there's some interesting topics in terms of how condensates play a role in development, evolution, and of course disease, which was talked about earlier. And then there are engineering and medical opportunities, as there always are. So that was a little bit about that. Then they talk a little bit about condensates and what they are. So as it turns out, the term biomolecular condensate is controversial. Some might consider all cellular structures to be condensates, since all structures comprise condensed matter. So that means that if you have like a material that has a mix of liquids and solids, that's a condensate technically, but that's not necessarily what others claim. Others might claim that nothing is a condensate since they define condensate as emerging through a phase transition, so they have a more of a functional definition than a structural one, which would require infinitely large systems to unambiguously identify them using tools from statistical mechanics. So, some people define a condensate as being formed by a specific process where you have a phase transition, and you can look at that phase transition through statistical mechanics and define where that happens. Other people just say, well, anything can be a condensate as long as it has a the structural properties that we expect. But this isn't really helpful for what we need to understand in the cell because, it's hard to measure biological condensates in the way that we might measure them in like a experimental physics environment. Whereas, just defining the entire cell as a condensate is not useful either. So people have developed a wealth of terms like aggregates, blobs, bodies, clusters, foci, granules, hubs, macromolecular assemblies, microdomains, nodules, puncta, ribosome exclusion zones, speckles, spots, and other things, although those are the ones that you might see in the biological literature to describe what we might put in this category of biomolecular condensates. So what they're saying here is that if you go back in the cell biology literature, people are looking at things under a microscope. They're trying to describe them. Sometimes they give them some sort of quasi-functional cast where they say this thing looks like something that looks like a granule or foci or a blob, and which are maybe suggest some sort of function that doesn't exist, or it's something that looks like something else in the world. And so it's hard to know kind of what that thing is, and these terms stick around, so it's hard to really kind of once we learn more about these structures, it's hard to really know what they do. Partially this is a problem of language where we have these terms that are maybe unhelpful, but also really kind of getting into the function. Do things that are described as blobs all behave the same? Or do all blobs behave the same as aggregates or granules or whatever? So, we don't really understand the good, all of the variation here. And it's a mistake to lump them all maybe into this category. On the other hand, they probably have very similar functions. Such a zoo of terminology with subtly distinct meanings hinders coherent communication and suggests that clarifying the concept of condensates themselves might be helpful. So, there's some reinterpretation there, but also just understanding what we mean by condensate is also important. So, all condensates really have the similar properties. Researchers tend to agree that condensates are compartments with very different composition from their surroundings. So it's like, you have like any sort of structure in a cell, it's compartmentalized, it has things inside of it that are different than the outside. And usually that's done to protect that structure from the environment. These surroundings are separated by a well-defined interface that is not established by a lipid membrane. Condensates are non-membrane bound compartments that can exclude membrane bound organelles, such as the nucleus or mitochondria, though sometimes they associate with one another. People also tend to agree that condensates have dynamic, non-stoichiometric composition, so individual complexes like the ribosome don't count. So, this is just kind of excluding different things in the cell that you might if you look under a microscope, you might misattribute it to a condensate. Moreover, condensates are typically described as having, at least in part, an amorphous internal structure. It's just a bundle of macromolecules in which the relative positions of molecules are not guaranteed, to exclude ordered structures such as amyloid-like aggregates and microtubules. So this is, again, where you see things sometimes that are microtubule bundles or amyloid aggregates, and those are not the same thing as what we're talking about. Finally, many people stress that condensates exhibit emergent properties. In other words, the properties of individual molecules differ from those of the collective. However, it's unclear at what size or molecule count such properties emerge. So how large does a condensate have to be? And in that sense, we don't have any hard and fast rules. It doesn't seem to be deterministic, so it's probably some sort of stochastic process of self-assembly, and then at some point they behave differently according to some sort of phase transition. So this is all kind of unknown or not clear to people. So there was this workshop at the KITP, which is in California. This was the Physical Principles Shaping Biomolecular Condensates workshop, happened in 2025. So this is their blackboard where they kind of laid out some of these ideas about what condensates are and what they are not. They had a whole bunch of things that they talked about. Thinking about everything from the structure to the function to the dynamical systems aspect of it. And then, of course, you have to go through all these defining terms, what to look for, what kinds of properties do they share, what kinds of things can be excluded. And in that, of course, you have a lot of heterogeneity. So that's what makes this a very hard problem. But there are a lot of interesting physics and a lot of interesting biology. So this is interesting. So there are also more controversial properties that are often discussed in the context of condensates. Some papers equate condensates with liquid-liquid phase separation, but it is often neither clear whether the involved objects have liquid-like properties or maybe also some solid-like properties, nor whether they actually form by phase separation. So sometimes things form by phase separation, sometimes they don't. Sometimes, or the formal process that we recognize, sometimes they have just liquid properties, sometimes they have some solid properties. As you can imagine, if you have these sort of just tangles or aggregates of things, it includes, sometimes it'll include all sorts of things in it. And so in any case, phase separation denotes the process, whereas the outcome is better described as phase coexistence. Phase coexistence is unambiguously defined only in terms of thermodynamically large equilibrium systems, so its validity in small living cells out of equilibrium is unclear. So sometimes we have these physical concepts that we were trying to apply to things like cells, and we have to be careful because they look like the thing that we see in physical systems, but they may not behave exactly in the same way. Finally, condensates are often associated with biological function, but some condensates may form without biological function. In any case, proving biological function can be hard, but it is still useful to discuss condensates even when their biological function is yet unclear, so we can have some grasp of what's going on with this structure. And so this rest of the paper, I think, really kind of goes through both the physics of condensates and the biology of condensates. And so if you're interested in that, that can help, clarify a lot of things. So figure 4 is the physical framework of condensation, describes how various control parameters affect condensate properties. So here they're looking at control parameters, of course, as a way to maybe manipulate condensates experimentally or use them for medical purposes or engineering purposes. So they define some control parameters, and then they have these condensate properties, and they're not quite sure how they all kind of interrelate, but they know that the control parameters should vary the condensate properties in certain ways that are, perhaps useful to engineers or to experimentalists. So control parameters include intermolecular affinities. We're talking about biochemistry, biochemical affinities, molecular concentration, what's the concentration of molecules, intracellular structures, physical fields and extrinsic stimuli, and then active processes in the cell. So these are all things that are contributing to condensates, controlling their size, controlling their shape, their material properties, and so forth. And then the properties themselves, their composition, what are they made of precisely, location in the cell, maybe they're close to the nucleus versus out near the membrane. That may be important. The kinetics or the timing of formation, the material properties, the internal organization, and the morphology, which is defined in terms of size and shape. So these are all kind of, this is building towards sort of a model of being able to manipulate condensates in a way that helps us to control them or understand their structure better. So these control parameters are the knobs, and they kind of go through the definitions here. They actually have some success stories, which is where we can understand some of the details of condensates that are understandable to science. So P granules is interesting because it comes from C. elegans. So among the first condensates described as such are P granules. And if you look at a C. elegans embryo, you'll see P granules in the images. And P granules are part of this P sublineage, which is the germ cell sublineage. So if you look at a lineage tree, you'll see that there are these founder cells, and then there's this lineage called P, and there's P1, P2, P3, and it branches off early, and it's basically where the germ cells get specified. So it's where the eggs and different components of the reproductive system get specified. And P granules are in that sublineage, and they play a role in germ cell specification. So this is, you'll see this in different microscopy images. P granules are initially distributed throughout the one cell embryo, which is where we have a fertilized embryo that hasn't made any cell divisions yet, but are ultimately inherited by the posterior daughter cell at division. So you have the one cell, it divides into AP and P1. The P1 is actually the posterior end of the worm, but it will become that P lineage, which is the germ lineage. So you have P1, P2, P3, and then by the time you get to P4, that's the germ lineage. So P granules are in this P part of the posterior part of the embryo, and then eventually they localize into the germ cell. So that's interesting because if we remember when we talked about these condensates, we didn't know where they were going, what role they played in terms of their position. We just kind of thought of them as stuff that's randomly floating throughout the cytoplasm. But now we know that at least in C. elegans embryos, they localize in certain cells that are dividing and ultimately get down to some sublineage. So, that's interesting because that suggests that the P granules kind of have a map of where to be during cell division so that when the cell divides, it's in the right cell, in the right daughter cell. Okay, so and then this asymmetric segregation, first described in 1982 by this citation here, 19, cannot be explained by cytoplasmic flows or localized degradation. Instead, Brangwyn et al. Proposed an alternative mechanism. Before cell division, P granules in the anterior half of the embryo shrink and dissolve, while those in the posterior half condense and grow. So they can shrink and they can grow, and in this case, they're doing this differentially. This novel description explained not only the asymmetric localization of P granules in the embryo, but also their fusion, dripping and wetting behavior in the adult gonad. And so that's one example. And then there's some examples from bacteria that's linked to the polar organizing protein Z or POP-Z microdomain. In sexually reproducing organisms, genetic diversity is generated through DNA exchange between homologous chromosomes during meiosis. Numerous double-stranded breaks are formed along each chromosome, but only a few of these sites become crossovers. Putative crossover sites interfere with one another, such as that mature crossovers are distantly spaced. So this was observed in Drosophila in 1913, where actually in looking at Arabidopsis thaliana, which is a plant, and the animal C. elegans, now suggests that the number and positioning of crossovers are determined by coarsening of recombination nodules. Recombination nodules are condensates that form at double-stranded breaks. Nodule components such as the ring finger protein diffuse laterally along the synaptonemal complex that hold homologous chromosomes together, redistributing from many small nodules to a few large nodules, which then promote crossover formation. So these nodules that occur during DNA replication and transcription. So even, this is something that happens in different places in the cell and not just, so condensates have very wide-ranging functions. And so, if we're going to create this sort of new category of biology, we have to keep an open mind. I guess the idea being that condensates have sort of certain similar physical characteristics, similar functional biological characteristics. So so that's the paper. I think that's actually pretty interesting based on kind of some of the things we're interested in with cytoskeleton and some of the things we're interested in with biological function in cells, and of course cell division, because this process relies on cell division quite a bit. So I was impressed with both of the presentations today. But did we have any other questions before we go?
No. Yeah. I wanted to ask if there's anything I could work on further about this.
Well, I mean, probably. I think some of the graphs you made were interesting. I would keep working. Maybe some of the graphs are just based on one cell. So if you can look across cells, use other cells.
Okay, so get the spatial context basically.
Yeah, I mean, because there are differences between cells, of course. We want to know if maybe other cells are behaving differently. In the lineage tree, you'll notice that not all, sometimes you have sublineages. You'll have things at the anterior end, you'll have things at the posterior end, and they're going to behave differently. So that's one thing. And then label what cell you're looking at. Are you looking at some AD cell or something in the posterior end of the embryo or what? Another thing was the one we talked about that graph that had the properties where the movements kind of clustered, and that might be cell division. We could figure that out if we knew what cell it was and we knew the timestamp. So if we could have the time, what specific timestamp is it? And then we can go look it up and say that's what's happening in that cell.
So basically, get a clear explanation on what is happening and why it's happening during that time step.
Yeah. So each cell each developmental cell that we're looking at in the data set has a certain lifespan. It divides from a mother cell. It lives for a while and then it divides, or sometimes it dies off. But that lifespan is basically finite. We're looking at basically it's a newly divided cell. It's migrating to a position, and then it's dividing again. When it divides again, then the daughter cells move somewhere else, and that process continues. So sometimes it makes sense to maybe track the eigenvalues for multiple cells that are daughters to one another. So you'll see if you look at the lineage tree, if you know where to find that, I can send you a link. You can find the daughters pretty easily, and they're always the same. So it's like in any data set, they'll always be the same, but you can see that kind of progression through the.
Okay.
So those are the kind of things to follow up on.
All right, I'll look into those. Yeah.
All right. So Susan, said there's a Journal of Biological Physics talk by Matthias Serra. Oh, okay, Matthias Serra. Yeah, I know that is. And that's UC San Diego.
It was about avian gas relation, and it was very good. It shows the actomyosin, where actomyosin gathers in the avian embryo as the neural tube is formed, the neural streak is formed. And it's interesting. You can see there's a loop of it this way and some on the sides. Like it's almost like it's differentiation waves. Where there is actomyosin, there is cell cramping or cell division. So it correlates. So that's an interesting lecture to watch.
Yeah.
And I noticed Dick wasn't there, so I think he should be watching it. But
I noted it down. Yeah.
Okay. Well, they just sent me a copy of where to find the lecture because it was recorded.
Yeah.
Would you like that or?
Yeah,
sure.
Yeah. Okay. Well, I'll send it to Bradley and you can distribute it.
Okay.
Yeah. Sure.
Unless I have your email. But
I can put in my email. Yeah.
Okay.
There we go.
Okay. All right. We'll get that and I'll send it to you.
All right. Thank you so much. Yeah.
Okay.
All right. See you next week.
All right.
Yeah.
Okay. Bye.
Thank you. Thank you, everyone. Bye-bye.